So, welcome everybody to the Aldrich commemorative lectureship today. Thank you for coming. This is, I believe, the 18th annual Michael S. Aldrich commemorative lectureship. And we try to make it annual. Actually, this one we started trying to schedule in August of 19, or that's when we started working on it, and it kind of got delayed by the pandemic. I just wanted to say a brief word about the history of the Aldrich lectureship. So Dr. Aldrich founded the University of Michigan Sleep Disorder Center, the first one in 1985 with the help of Dr. Sid Gilman, who was chair of neurology at that time. And he, over the next 15 years, he really established it as a national leader in a very early field. He was remarkable for his ability to attend to and develop in such a fine fashion the tripartite mission of the center in terms of clinical work and his own clinical expertise and research that he got started, particularly in narcolepsy and sleepiness and education, starting one of the very early fellowships. And so we have this lecture each year in his honor and give an award. We are very pleased to be doing that today. We have a stellar lecturer to receive the award and give the talk today. And I'm going to ask Dr. Garwood, who was kind enough to work on the arrangements with Carol Kuhlman and April Gallant and host our speaker, to introduce our speaker.
Thanks, Ron. So I know many of you know Dr. Holtzman and have worked with him and count him as a friend and a colleague. And we're really just delighted to have him here today. Just to go over his background, he received his Bachelor of Science and Medical degree from Northwestern University, followed by a neurology residency at UCSF from 1985 to 1989. He did postdoctoral research at UCSF from 1989 to 1994. There, in addition to his research, he founded a memory disorders clinic. He moved to Washington University in 1994 as an assistant professor to both start his own lab as well as to get involved in clinical activities and teaching. And he's currently the professor and department chair at Washington University in St. Louis. There he's also the scientific director of the Hope Center for Neurological Disorders and the associate director of the Knight Alzheimer's Disease Research Center. He and his lab's accomplishments include showing in part how ApoE4 contributes to Alzheimer's disease, the development of a method to measure protein synthesis and clearance in the CNS of animals and humans, development of CSF biomarkers for Alzheimer's disease, and demonstration of how synaptic and neuronal activity and sleep affect amyloid beta and tau levels dynamically in vivo acutely and chronically, as well as development of anti-amyloid beta antibodies and helping these antibodies make their way into phase 3 trials for Alzheimer's disease, and anti-tau antibodies and helping them make their way into phase 2 clinical trials. He's received a number of honors, including being a recipient of a Paul Beeson Physician Faculty Scholar Award in Aging Research, the prestigious Potemkin Prize from the American Academy of Neurology for Research on Alzheimer's Disease, the MetLife Award for Alzheimer's Disease Research, a Merit Award from the NIA, election to the National Academy of Medicine of the National Academy of Sciences, election to the National Academy of Inventors, an Alumni Merit Award from the Northwestern Feinberg School of Medicine, being appointed to the National Advisory Council of the NINS and NIA, the Chancellor's Award for Innovation and Entrepreneurship, and the Carl and Gertie Corey Award from Washington University. He was elected Fellow of the American Association for the Advancement of Sciences. He has served as the past president, or is serving as the past president, I should say, of the American Neurological Association. He's trained 50 or more graduate students, postdoctoral fellows, and physician scientists, many of whom have gone on to successful careers in academia and industry. His accomplishments are many, and really, when I read his biography and look at what he's done, he's really been an exemplary clinician, educator, and researcher. Many of those same qualities Michael Aldrich also had. He's certainly had a big impact in our field of neurology as a whole, and certainly with Alzheimer's disease, but more recently in sleep medicine and the intersection between the two. And we're really excited to have him today to be our Michael Aldrich speaker for our 18th annual commemorative lecture. And his talk is titled Sleep and Alzheimer's Disease, the Bidirectional Relationship with Amyloid Beta and Tau.
Well, thanks so much, Dr. Garwood and Dr. Chervin. It's a real honor to give the 18th Aldrich Lecture. And it's also wonderful to see so many colleagues and friends on the Zoom screen. Having said that, I'll switch the screen over to my talk now so that, let me just get this share screen and then we'll get going. Just a second. Everybody see that okay? All right, good. Great. So what I'd like to tell you a story about today is something that really my laboratory stumbled into about 13 years ago, but has now become a major focus of research in our group to better understand the relationship between sleep and Alzheimer's disease. And not just the fact that there are sleep abnormalities and circadian abnormalities in people who develop the disease, but how the sleep-wake cycle may be involved in the actual causation of disease or contribute to the causation. Let me, the disclosures I have are listed here. The only one that would be relevant for what I'll directly talk about today is just consulting for some companies where they're investigating different aspects of new sleep treatments. All right, so this is also Neurology Grand Rounds, and so I thought I would just give a very brief background about Alzheimer's disease, and then get into the relationships between sleep and some of the proteins that are involved in the disease. So dementia, as you all are aware, dementia is a decline in memory and other cognitive abilities that is sufficient to impair social and occupational functioning. And the prevalence of dementia overall is around 2 or 3% at age 70, but by age 85 and over, it's around 50%. And obviously, there's many different diseases and disorders that can contribute to dementia. Alzheimer's disease probably contributes to about 70 to 75% of cases, and it's one of the most, if not the most expensive disease, at least in terms of dollars to care for individuals and their families that we have right now. This is one of the reasons you're probably all aware that the NIH has increased their budget to try to cure Alzheimer's and related disorders by about almost 8-fold over the last 6 years. So the clinical features of this disease is there's very, very gradual onset and progression. Usually the first clinical deficit is in recent or episodic memory, but at the same time that this begins, there is almost always some other cognitive dysfunction as well, such as executive dysfunction as seen with difficulties with problem solving and also attention. Language is usually impaired, especially trouble with coming up with words. That can be the first symptom in some cases. Visual spatial dysfunction is frequent as the disease progresses. And there is behavioral dysfunction from the beginning, often apathy, but as the disease moves forward, depression is frequent, and sleep disruption is also quite common. There's two major types of Alzheimer's disease, the so-called autosomal dominant form, which has been very informative about trying to better understand the pathophysiology of what's going on. And this accounts for less than 1% of total cases, but it's certainly led to a lot of insights into both understanding the diseases, developing new, as well as developing new treatments. The late onset form of disease is usually after the age of 60, and this accounts for most cases of the disease. Despite the fact that the autosomal dominant form is directly caused by particular gene mutations, the two strongest gene mutations or two strongest risk factors for late onset Alzheimer's disease are age and family history. And in fact, of the common diseases in humans, Alzheimer's disease is one of the most strongly genetic. The two major, there are several pathological features you see in the brain of somebody that, at least in regard to the specifically Alzheimer's disease that you see, which is the accumulation in the extracellular space of the brain of these amyloid plaques, which are made up of the amyloid beta peptide. Normally, this peptide is produced from all cells, but high levels by neurons, and it's in a monomeric form that's produced and cleared rapidly by all of us all the time. But as this disease develops, it begins to aggregate. The other major protein that aggregates in Alzheimer's disease is tau, which is an intracytoplasmic protein shown here in blue, and it aggregates in cell bodies and dendrites and is also a very important feature of the disease. The other feature of the disease, which is receiving a lot more attention now than previously, is the inflammatory response. So there's a strong astrocyte and microglial activation, which I think for many used to think that this was kind of a secondary phenomenon of the disease, and it's very clear that this innate immune response is involved in pathogenesis. I think one of the most important realizations about the disease that has come about over the last 30 years, but has been solidified with the advent of biomarkers where one can track what happens to pathology over time in living people, is that this disease, if somebody presents with the clinical manifestations of disease with so-called very mild AD or MCI due to Alzheimer's disease, that the disease at that standpoint from a pathological perspective has already been going on for approximately 20 years. And the amount of amyloid accumulation in the neocortex is already close to its maximal extent at that point. Neurofibrillary tangle accumulation it's on its way up in certain parts of the neocortex. And amyloid plaque deposition begins in the neocortex, as I was just mentioning, around 15 to 20 years before the onset of symptoms. So if you're trying, the reason I point this out, not only because it's probably relevant for developing preventions or delays of treatment, delaying progression of disease, is that in terms of understanding the pathophysiology, if you want to understand what contributes to the start of this pathology, we probably need to start before this pathology begins and use model systems in humans to understand what leads to the accumulation of these abnormalities. So this is a complicated slide, but for many years, one of the theories of this disease has been called the amyloid hypothesis. And it basically stated that as the amyloid beta peptide aggregates in the brain, that it ultimately leads to several downstream events that leads to the disease. And I think it's much more complicated than that. I think it's clearly the case that the deposition of this peptide in different forms is necessary for Alzheimer's disease, but it's probably not sufficient. We need a number of other things to occur, such as the A beta can somehow drive the accumulation and spreading of tau pathology in the brain. And there's this inflammatory response is very involved in modifying both A beta and tau and its relationship. And I'm not going to go a lot more into that component in this talk, but one of the things that I do want to talk about more is why does the A beta peptide aggregate in the first place? And one of the things that we do know is that the mutations that lead to autosomal dominant Alzheimer's disease that are in genes like presenilin 1, presenilin 2, or the amyloid precursor protein shown here, what all of the different mutations in those genes do is they mostly elevate either the amount of this A beta 42 peptide or the ratio of A beta 42 to 40, which over a long period of time, because this peptide is aggregation prone, tends to drive this reaction earlier in life. So for example, in people with presenilin 1, presenilin 2, or APP mutations, instead of developing this pathology, let's say at age 60 and then going on to get clinical disease at 75, for example, these patients will develop A beta deposition beginning in their 20s and then become sick cognitively in their 40s often. But in the late onset form of disease, there's not good evidence that it's marked overproduction or a higher ratio of this peptide that drives disease. But there is evidence that alterations in clearance of this peptide are involved, especially with, for example, how ApoE might influence risk for disease. And one of the things that I'll talk about more in this talk is that one of the things that our group and others identified about 15 years ago is that one of the things that influences the levels of this peptide A beta in the brain is synaptic activity itself. And what I'll get into in the next after this is that the sleep-wake cycle seems to directly influence the synaptic activity to influence A beta generation as well. So the initial experiments that led my lab to sort of look at synaptic activity first in Alzheimer's disease were sort of driven by the kind of observations shown here. And this is work from Randy Buckner and Mark Rakel and Mark Minton back in the early 2000s, where they basically, when amyloid imaging first became available and you could determine where in the brain the amyloid beta peptide was depositing in living people, if they looked at people who were still cognitively normal but just developing amyloid deposition, you can see this pattern of A beta deposition in red, where it's kind of in the lateral temporal parietal region, part of the frontal lobe, and then the posterior cingulate precuneus region of the brain. And when they saw that observation, they noted that this distribution of pathology that started early overlapped a lot with what they had termed, what Mark Rakel and others had termed the default mode network in the brain, which are areas in the brain that over a 24-hour period appear to be the most synaptically active if you average it out over a long period of time. And so the question I wondered about was, well, why would those regions be affected with amyloid deposition first? And one possibility is that if synaptic activity is highest there first, how might that be related to development of this pathology? And I'll just summarize a whole series of papers that we published over the years, where what we found is that, and this was started by John Cerrito, then a graduate student in my lab, where what we found is that if you study synaptic activity and its relationship with A beta release by neurons, that as synaptic activity increases, that stimulates the re-endocytosis of synaptic vesicles, and that also re-stimulates the endocytosis of the amyloid precursor protein, which is not only at the synapse, but is enriched at the synapse. And when this happens, it drives endocytosis of APP, and that is where A beta is generated in endosomes, and then the endosomes come back to the plasma membrane and fuse and release A beta. So basically, increases in synaptic activity locally will lead to an increase in release of the A beta peptide very dynamically in the brain. So some of the experiments that we also did to look at this further were that if you look in the mouse brain, for example, and you ask where is A beta levels, where are they the highest? And these experiments were all done by microdialysis to measure the peptide in the extracellular space of the brain. We found that endogenous levels of A beta in different brain regions correlate with regional neuronal activity in those regions and predict, if you follow in an animal model that develops amyloid deposition, they predict subsequent levels of amyloid deposition in those brain regions. Then it's very simple physiological manipulations like stimulating the whiskers in a mouse, which stimulates neurons in the whisker barrel system, that increases the A beta peptide. And if you cut the whiskers and decrease neuronal activity, physiologic neuronal activity, that decreases A beta levels. So those are some of the experiments that we were doing at the time when we happened to stumble upon a potential relationship with sleep. And Jae-Eun Kang, a very talented graduate student, was doing experiments to try to understand what physiological events, at least at this time in the mouse, that were undergoing different behavioral manipulations, how did those behavioral manipulations influence the A beta peptide? And what she tended to notice in the course of these experiments is that the A beta peptide, and this is in mice, was highest during the dark phase, and mice are awake more during the dark phase, and they sleep more during the light phase. So you can see the A beta peptide is higher during dark, lower during light, and this just keeps going up and down over several days that you make these measurements. And the A beta peptide is related to the amount of minutes awake. Now, in these experiments, we could only measure the peptide every hour, so it wasn't fine manipulations. In more recent experiments in John Cerrito's own laboratory, he finds that when an animal goes awake or asleep, literally over a minute or two, the peptide will vary over the very short periods of time. So, and this has also been observed in human cerebrospinal fluid. In this study carried out by Randy Bateman's lab, they were measuring these humans he was studying had lumbar catheters in place, so kind of like an IV, but in the CSF space. And if he sampled the A beta peptide over a 24-hour period, you could see and average this over many people, you can see it's higher and lower. And this tends to correspond with when people are awake, it's highest, and sleeping lowest. So one of the things that in a subsequent study after Jae-Eun's paper came out, Adam Biro, another graduate student, was looking at, again, the levels of the A beta peptide during the light phase when the animals are sleeping more versus the dark phase when they're awake more. And again, he could see there's about a 30% difference in the levels of the peptide depending on the time of the day that he was sampling. But what's also really interesting is that he also measured the levels of lactate. And one of the things that we know from in vitro and in vivo studies is that the levels of lactate directly reflect the synaptic activity levels in the region of the brain that you're sampling from. So you can see that when animals are more awake, lactate, again, just like A beta, is about 35% higher than it is when the animals are sleeping. And in work by other labs that we've also replicated is that if you put a probe into the brain that can rapidly measure metabolites like glucose or lactate, is that when an animal goes from wake to sleep, literally in 1 second, lactate will go up when it gets awake and it will go right back down as soon as it's sleeping. So this is a very rapid dynamic change that occurs in the brain, probably simply reflecting differences in regional synaptic activity that differ between awake and sleeping animals. So once we had first made this observation that the A beta peptide cycles with the dark and light cycle, we wanted to understand, is this due to changes in sleep, wake, or other potential circadian changes, for example? So one of the first experiments that Jae-Eun did was she gave orexin, which is specifically promotes wakefulness. And so you can see that the A beta peptide is, again, it's higher during the dark phase. But when she gives orexin, the peptide should continue to go down. But when you administer orexin, in this case in the CNS, it keeps the peptide higher than it would be otherwise, and the animals are awake more. She then did the converse experiment and gave an orexin receptor antagonist. And normally during the dark phase, the A beta peptide would continue to go up. But you can see here that it stays lower. And that the animals also sleep more, as is known when you give a dual orexin receptor or an orexin receptor antagonist. Miranda Lim then was a neurology resident at WashU, and she did several months of experiments in my lab at that time. And we wanted to ask whether if you chronically affected sleep, would that influence, all the measurements I was just showing you are the monomeric A beta peptide, not the aggregated form. So we wanted to do experiments where we either sleep deprived or promoted sleep over longer periods of time and see if that affected the deposition of the pathological form of the molecule. So in these experiments, she sleep deprived, in this case, one of the animal models that develops amyloid deposition called PSAPP mice. She did this in 2 different models. I'm only showing you one here. where if you start sleep depriving the animals for about a 6-week period, right when they're about to develop amyloid deposition, it increased pathology by about 2.5-fold in multiple brain regions that she examined. She also did the converse experiment where she gave this orexin receptor antagonist, almorexant, again, right before animals develop amyloid deposition, and this suppressed amyloid deposition substantially. So these experiments overall, while this acute regulation of the A-beta peptide was observed, it's possible that this chronic change, the sleep deprivation or sleep promotion, may also be affecting this kind of longer term pathology by the mechanism I just showed you, or it could be some other mechanism that we don't yet understand. Now, a few years after we published this paper showing that the A-beta peptide fluctuates with the sleep-wake cycle, experiments began to be published by Macon Niedergaard's lab, where she and her colleagues came up with the term the glymphatic system that has received an incredible amount of press, which basically argues that there is a fluid flow within the interstitial fluid of the brain that goes from the arteries to the capillaries to the venules, and that fluid flow regulates clearance of molecules in the CNS out of the interstitial fluid into the CSF space. And the data that she's when she first published a paper about this theory, one of the molecules she tested to see if it had an altered clearance when she in the brain was the A-beta peptide. So in this top experiment, what she did is she took iodinated A-beta peptide, injected it into the brain interstitial fluid, and measured its clearance from the brain. And you can see that in animals that were awake versus those that were sleeping or under anesthesia had slower clearance. Whether this phenomenon contributes to some of the effects that I was showing you, we're not sure. But what hasn't been shown in regard to this theory is whether the glymphatic system is influencing clearance of endogenous molecules made in the brain. So we don't really know how this truly contributes yet or not to these alterations that are occurring rapidly in the interstitial fluid. It's certainly possible that contributes, but we don't know yet. So one of the things that we wanted to understand after we made some of these findings is that given that sleep disruption and sleep promotion in animals acutely influences A-beta, does this actually occur in humans? I'll just show you one study that was recently published by Brendan Lucy, who is the head of our sleep section in one of my former mentees. He's been doing studies where he has also been using lumbar catheters in humans to study the metabolism of different proteins as influenced by sleep. And so he did this study where he took healthy volunteers, they had lumbar catheters placed, he sampled CSF every 2 hours from the catheters. All the subjects had a polysomnogram during these studies, and then some of them were behaviorally sleep deprived and some were not. And so if you focus your attention to these top graphs, what Brendan found is that in the red line, these are individuals who were sleep deprived overnight. And you can see that by measuring in this case, the A-beta peptide by mass spectrometry, you can see there's about a 40% increase in the levels of this peptide in human CSF in those that are sleep deprived versus those that are not. The other thing that Brendan did, which I think is a really important experiment, is that he metabolically labeled these people with C13 leucine intravenously, which is allows you to do a pulse chase experiment to determine the rate of production and clearance of the proteins you're measuring. And by doing, you can see that the fractional synthesis and clearance rate does not, the levels of these labeled peptides is not altered between the different conditions. And in doing mathematical modeling, what would happen is if clearance of the peptide was altered, these curves would differ. And basically the A-beta curves for the red line would actually divert quite a bit from the other lines if clearance was affected. If synthesis was affected, you would see no change in the slopes of these curves compared to each other. So this strongly argues that the effect of altering A beta levels by sleep deprivation is due to altered release and synthesis, not an effect on clearance. To further assess whether these effects in humans of altered sleep are specifically due to altered slow wave sleep, Yoel Ju, who's an associate professor at WashU, also in our sleep section, did experiments where she created a system where individuals would come into the sleep unit and wear a device over their ears where they would and have a polysomnogram when they would basically, as they would go into different stages of slow wave sleep, the system would start making higher and higher noises and wake them up, not enough to wake up completely, but get them out of slow wave sleep. And by doing this, she also did a spinal tap in the next morning and found that the more that slow wave power was altered, the higher the A beta peptide level, arguing that it's the change in slow wave sleep which is driving this change in the A beta peptide. And then finally, in this first part of the talk, I wanted to just go through, there is some ideas actually from humans that perhaps sleep maybe, if you had something that affected A beta production or release, in this case, I think sleep disruption is doing that, but you also had something that affected clearance of A beta like ApoE, which I won't go into here, but we've shown that ApoE influences A beta clearance and E4 slows that clearance more than the other isoforms, then maybe there would be a dual effect of sleep deprivation and ApoE4. And there's some evidence from human studies that this may be the case indirectly. So in some recent studies, we've been trying to see if there's any linkage between ApoE genotype and sleep and its effects. So this is a new sleep deprivation system that Cheneng Wang, a postdoctoral fellow, set up in the lab, where basically we can, in a much more automated way, disrupt sleep for different periods of time in different ways, and we can monitor the mice remotely or in person. So we did an experiment where we took APP mice that develop amyloid deposition, except these mice were crossed to mice that express human ApoE4 or human ApoE3. And then at 5 months of age, these mice were sleep deprived or not for 2 months. And then in this case, what Cheneng is measuring is the amount of A beta plaques, basically A beta plaques in the brain. And if you focus on the hippocampal data, you can see that in the E4 mice controls, there's a certain amount of amyloid deposition. And in the sleep deprived mice, there's about a doubling of amyloid deposition. But what's interesting is that we don't, in the ApoE3 mice, there's a subtle but not significant effect. So it looks like the effect of sleep deprivation is much stronger in animals that express ApoE4 and ApoE3. And we're now in the process of trying to better understand the mechanism that's underlying this change. Okay, so I've been talking all about now is how if you have altered sleep or wake, how it affects the levels of the A beta peptide and its deposition in the brain. But what about if this peptide aggregates in the brain? Does that in and of itself cause abnormalities in the sleep-wake cycle? So a few years ago, Ji-Hoon Roh, a neurologist postdoc in the lab, did these experiments where he took one of the models of amyloid deposition, PSAPP mice, and this is just showing you the time course of amyloid deposition that occurs in this model, so that by 9 months of age, there's a fair amount of amyloid pathology that occurs. He then assessed changes in sleep in this model, and what he found is that if you focus on the light phase when the animals should be sleeping more, is that as they develop more pathology, their amount of wakefulness increases from 20 minutes per hour to over 30 minutes per hour. So it's basically somehow this change in time is linked with an increased wakefulness. So one of the issues, though, in terms of causation is that these mice overexpress a variety of full-length proteins, not just the amyloid peptide. So we wanted to understand, is it truly amyloid deposition in some way that's leading to this change? So we used a technique which had previously been published many years ago, where if we vaccinated the mice subcutaneously with amyloid starting at a young age, this can virtually completely prevent the pathology from, amyloid pathology from developing. So we decided to vaccinate a group of mice or vaccinate them with a control PBS and see over time whether this would not only affect amyloid deposition or not, but whether the treatment would attenuate the changes in sleep or not. So what you can see in this experiment, we actually assess sleep in these mice at 9 months of age when there would otherwise be a lot of amyloid deposition. And then the PBS vaccinated mice are the controls. You can see that sleep is very disrupted. The amount of time that they are awake during the light phase is about, again, it's increased by about 15 minutes per hour. That's a big, big change. But in the mice that were vaccinated, they have a normal sleep-wake cycle in terms of the amount of sleep and wake you'd expect a mouse to undergo. So that strongly argues that this change in sleep, at least in this model, is due to amyloid deposition. It's not due to simply overexpression of the transgene. So what about evidence in humans that amyloid deposition may lead to disruptions in sleep and non-REM slow waves? And there's now a number of papers published from Yoel Ju at WashU, Adam Spira at Hopkins, Bryce Mander and Matt Walker at Berkeley, who've all shown that in humans, if you look at people that have amyloid deposition prior to when they're cognitively impaired, there are already elements of sleep that you can find that are abnormal, such as sleep quality, self-reported sleep, as well as non-REM slow waves. So I get this sort of supports that what we're seeing in mice appears to potentially be occurring in humans. Okay, so we've been talking about sleep and one of the pathological features of Alzheimer's disease, A beta, but what about the other major one, tau? So as I showed you earlier in the talk, in terms of the time course of Alzheimer's pathology, tau doesn't start to accumulate until just before, at least in the neocortex, until just before people become impaired. And there's many studies, I'll show you in a moment, some of them that strongly argue that it's where tau pathology is accumulating, which is where people develop the cognitive abnormalities that you see clinically. And actually, it's tau pathology that is in some way driving the actual neurodegeneration that we see, not just in Alzheimer's disease, but also in primary tauopathies. This is also illustrated in the dominantly inherited Alzheimer network patients that have been followed in a multi-center study, where if you look at different biomarkers for Alzheimer's disease in this population, it's very similar to what you see in late onset disease, that amyloid deposition starts about 20 years before pathology. But if you actually look at where tau pathology starts in these patients, it doesn't start till right before symptoms begin. And it also strongly correlates with the clinical abnormalities that you see in patients. Oh, so this is a study done by Keith Johnson, Risa Sperling, and colleagues, where they looked at late onset Alzheimer's disease or controls and asked also where does tau pathology begin to occur and does it correlate with cognitive abnormalities? So in these cognitively normal people, A, B, and C, this person has no amyloid deposition and no tau pathology. This individual has amyloid deposition. These two both have amyloid deposition, but what's shown here is the tau pathology on a tau PET scan. You can see a little bit starting to occur in the hippocampus and a small amount in the temporal cortex. But as people progress to mild cognitive impairment due to Alzheimer's disease, you can see that tau accumulation is now occurring quite extensively in the temporal lobes, and then as people progressed to more advanced stages of abnormality, that the tau pathology progresses to other areas of the neocortex. So this is, again, circumstantial evidence that it's the tau pathology that's correlating with the clinical abnormalities in people. This is a very recent study from Gil Rabinovich's lab where he's followed individuals who have amyloid deposition and then followed them, tau pathology and amyloid deposition, and follows what happens to the patient clinically. And you can see here in the green, what's on the x-axis is the baseline tau pathology, and then the y-axis is longitudinal brain atrophy, and there's a strong correlation between tau pathology and longitudinal atrophy, and that's not the case for amyloid deposition. So again, it's just argument that amyloid deposition is a trigger for several other aspects of the disease, including tau pathology, and it's the tau pathology that's linked with damage. So given all this, it turns out we began to study different aspects of tau metabolism several years ago, and despite the fact that tau is a very highly abundant protein in the neuronal cytoplasm, it also, like many proteins inside cells, is released outside of cells through a mechanism that we don't yet understand. And in these studies, Kaoru Yamada, a postdoc in my lab, was studying what regulates extracellular tau levels. And in this experiment, she was doing microdialysis in animals, in mice, and she did a simple experiment causing high potassium to be present, which causes a lot of neuronal activity locally. And that, you can see, strongly increases tau in the extracellular space by more than 2-fold over just hours. She also did experiments to increase neuronal activity, but for example, by giving NMDA into the brain, but at very low levels, not enough to cause seizures, but just increase activity. And you can see this increases tau by about 2-fold over just hours. This is an experiment by Karen Duff's lab then at Columbia University, where she took a model of tau pathology and she optogenetically stimulated one side of the brain and then measured tau pathology accumulating in both sides of the brain. And you can see in the stimulated animals over a month that there's increased tau pathology that occurred. Again, so it's kind of perhaps a similar story with A beta, that if you increase activity, both acutely and chronically, it increases tau release and tau pathology. So that led us to do several similar experiments regarding sleep with tau, and these studies were led by Jara Holt and Sarah Frischi, very talented postdocs, and in a collaboration that we did with Beth Israel, which I'll get into in a moment. So if you focus on this top panel, what Jara was doing at the time is she again was doing microdialysis studies. And if you look at these black dots, this is the level of tau in wild type mice during the light phase in black and then during the dark phase. And you can see that tau increases about 2-fold when animals are awake more. She then did the same experiment where she started measuring tau and then caused manual sleep deprivation. That increased tau more than 2-fold. She then did manual sleep deprivation in the presence of local tetrodotoxin in the hippocampus, and that blocked this change. So that suggests that tau is increased with wakefulness, and that increase is driven by synaptic activity. In collaboration with Brendan Lucy, who had already collected a lot of lumbar catheter samples from humans who had undergone sleep deprivation or not, Brendan measured tau in the CSF by mass spectrometry and found that in patients that were sleep deprived, there was about a 70% increase in tau levels. There was also an increase in other disease-associated proteins, in this case, alpha-synuclein, that was in the CSF space as well. And there's a very strong correlation between proteins that are increased with sleep, like A beta and tau, to each other. Now, when we saw this, I said, well, is this true for everything, or is it just some things? And so we also looked at other proteins that were in neurons or in glia, but were not enriched at the synapse. So it turns out A beta APP is enriched at the synapse. Tau, actually, it turns out, is enriched at the synapse, as is alpha-synuclein. But when you look at other proteins that are highly present in neurons, like neurofilament light chain, when we sleep deprived humans, we saw no change in neurofilament light chain in the CSF. We also saw no change in GFAP, an astrocyte protein. We're trying to better understand now if these changes are again, they're sort of following this pattern that things that lead to increased wakefulness are increased if they're enriched at synapses versus not. And that's something that we're doing in a more unbiased fashion in current experiments. So again, like in the amyloid experiments we had done, we also wanted to know if we chronically sleep deprived animals, would this increase tau pathology in the brain and what's called tau spreading. So in a lot of disorders where proteins aggregate, like in prion diseases, the proteins tend to be in one area of the brain, and then they tend to spread to other areas. So in order to model this for tau, we injected aggregated forms of tau into the hippocampus of a tau model that would then, the seeds would then cause this local tau pathology, as you can see here in panel F. And so what we did is we seeded these tau mice with aggregated tau to cause tau aggregates or tangles, in this case in the hippocampus. And at the same time, right after we did the seeding, we then sleep deprived animals for 1 month and then assessed the tau spreading in the brain. So you can see there's no effect on the amount of tau that was seeded, but we didn't even start doing the sleep deprivation until after the seeding had already taken place. But what you can see is that in areas where the tau is spreads to the synaptically connected brain regions, such as the locus coeruleus or the entorhinal cortex, you can see that in the animals that are sleep deprived, there was about a doubling of tau spreading that occurred from one area of the brain to another. And again, whether this is due to this acute effect on tau release, then leading to more spreading, or some other mechanism we're still trying to understand. We then wanted to ask a final question about this, is whether, are these changes in sleep-wake really that we're inducing by actually, manipulating the animal behaviorally, like keeping it awake by stimulating it or other means? Are these changes really due to effects on sleep-wake versus, let's say, stress or other things that we're inducing by keeping the animals awake? And so we embarked on a collaboration with Chris Saper and colleagues, Cliff Saper and colleagues at Beth Israel Harvard, where Cliff had for many years has been studying the different nuclei in the brainstem and other regions which influence sleep-wake. And one of the areas that was shown to do this is the supramammillary nucleus. And so what Cliff's lab, led in this case by Nigel Peterson and Patrick Fuller, had done is they had used a channel, a DREADS channel, which is a channel shown here called HM3DQ, which if you put this into neurons, and you can stimulate these channels with an otherwise innocuous drug that will not affect neurotransmission in other parts of the brain. So what they had already done is to use this compound CNO to stimulate this channel in the supramammillary nucleus, and shown that if you do that, you can cause a mouse to go from basically being awake about, let's say, 1/3 of the time to being awake 100% of the time for about 8 straight hours, which is incredibly abnormal. A mouse is never awake that much for that long a time. And in fact, what's really cool about their findings is that when an animal was stimulated to be awake like that due to this neural manipulation, it didn't cause any other changes in the behavior of the animal. They weren't stressed, they behaved normally. And so we thought this was a nice system to try. And so Sarah Frischi went to their lab, learned this technology where you can put this channel expressed under the control of a virus into the supramammillary nucleus. And if you do this in a mouse that expresses Cre in glutamatergic neurons, then you can turn on this channel that can be allowed to be turned on if you give the animal the chemical CNO. So we basically set up a system where we're able to do microdialysis and assess sleep while we would either give CNO to cause wakefulness or not. This is just some of the surgeries that were done to accomplish the experiment. So what Sarah showed is that if you look at this panel D, you can measure the levels in this case of wakefulness. And you can see that under normal conditions, the animal during the light phase was awake about 40% of the time. But when you give CNO, it's awake virtually 100% of the time for multiple hours in a row. And then it goes back to a normal sleep-wake cycle. Because you only activate the channel when you do this for, let's say, several hours in a row. So after accomplishing this, we then wanted to ask, well, what does this do to the other molecules we've been measuring? We also measured in these experiments stress hormones, and there's absolutely no change when we measure them. So if you measure the A-beta peptide when we give CNO and cause this increased wakefulness, as we had shown before with manual sleep deprivation, there's a very large increase of about 40% in the A-beta peptide. That's accompanied by an increase in lactate, which would correlate with the increased wakefulness. We then do an experiment where we measured tau in the same way, and you can see that tau increases about 70–80% when we stimulate wakefulness with this method. So I think all of these data, I think, are arguing that somehow the generation of these peptides and their release may, in fact, over time, if they're chronically altered, influence two of the primary pathologies in this disease. And just to follow up on the tangle pathology, we also wanted to know if tangle pathology also led to sleep abnormalities. And this is a study recently published by Brendan Lucy collaboratively with our lab, where basically Brendan's been following a number of individuals in our Alzheimer's disease center that are either cognitively normal or very mildly impaired. And you can see in this study where we had both CSF in many of them and also amyloid and PET tau imaging, that about 80% of the people that he's following are cognitively normal, and 20% or so are very mildly demented due to underlying Alzheimer's disease. And so all these patients have all these Alzheimer biomarkers done, but Brendan's been following them longitudinally with home sleep studies where with a device called the sleep profiler, where we can assess non-REM sleep as well as sleep power. And what Brendan found is that if you look at tau pathologies in these individuals, that non-REM slow wave activity is strongly linked with the tau pathology that's occurring in the brain when you correct for multiple other factors. And this is the distribution of the tau pathology and how it's linked to slow wave activity during non-REM sleep. What's interesting is that when he tried to correlate the amount of amyloid pathology with the same sleep abnormalities, there was a trend, but when you correct for all of the different variables, it's not as strong as what's correlating with the tau pathology. So arguing that the subtle changes in sleep that we're seeing in sleep power appear to correlate stronger with tau than amyloid. So to summarize, I think what many of these studies are showing is that fragmented sleep can lead to excessive A beta release. It could potentially alter the risk for amyloid deposition. This could further injure parts of the brain to lead to further abnormalities in sleep. This also appears to be the case with tau. And potentially this might lead to more tau pathology. Now, this is all still, I would say, a hypothesis. This isn't proven, but I think it's something that we're still trying to further address, and I think has important implications for how sleep-wake may interface with, in this case, Alzheimer's disease. So to summarize the talk, I think sleep-wake changes in endogenous activity regulate ISF, amyloid beta, and tau, and this ultimately appears to determine the likelihood by which these peptides will aggregate in the brain. And the recent data with stimulation of a wake center suggests these effects are via influencing sleep-wake, not via other linked mechanisms such as stress. And that A beta and tau buildup in the brain may lead to sleep disruption. This may occur during the stage of pre-symptomatic Alzheimer's disease. I think some of the future questions that we and others are addressing are, do other genetic risk factors for Alzheimer's disease, such as ApoE genotype, influence the effect of sleep on Alzheimer's pathogenesis? So I showed you some preliminary data suggesting in the animals you might be able to see something regarding that. Some of the things I think are important to understand in general, not specifically only for disease, are that what are the intracellular pathways that enables release of cytoplasmic neuronal proteins, such as tau and other molecules, into the extracellular space? And how do differences in neuronal metabolism that occur during wake and sleep influence this process? I don't think we understand much about that at all. Are there other pathways by which sleep disruption leads to increased accumulation of misfolded proteins, like inflammation, like autophagy, et cetera? Are midlife sleep disorders associated with the earlier onset of Alzheimer's pathology, synuclein pathology? And what ultimately might that put one at higher risk for really developing these disorders of protein aggregation? And I think given the data that tau pathology and markers of Alzheimer's pathology are related to quantitative changes in slow wave activity and non-REM sleep, can these changes be utilized as biomarkers that predict what's going to happen to people over time, and would they respond to different treatments? And then finally, can one manipulate sleep-wake in some way or other as a treatment or prevention for Alzheimer's disease? That's a very exciting area that I think we'd like to really continue to explore and are exploring at this point. So I will stop there. I tried to thank many of the people involved in these studies along the way, but wanted to leave time for some questions. I'll stop sharing.
Well, that was a phenomenal presentation. Thank you very much for outlining that story. We do already have 2 questions in the chat box. Peter, do you want to ask yours? Well,
looks like Peter Todd. I see his question. If you want, I can read it.
Yeah. Okay.
Peter asks, have you looked in animal models or in people how ApoE status influences tau and its relationship with sleep? That's a question that we're actually doing experiments on right now in animals. There is one paper published by David Bennett's group in JAMA Neurology, where what they did was they looked longitudinally in their large cohort of people that have been followed over periods of time and then go to autopsy. And what was found is that people that had better sleep characteristics that were ApoE4 positive progressed slower than those that were that did not have as good of sleep characteristics during that longitudinal follow-up, and that also was related to tau pathology. So that's some of the things that we're trying to address in animals are more of a cause-effect relationship to follow up studies like that result. Do you want to read the comments as questions?
Sure. Next, well, if the person's not, is Andrew Harley shoot here?
Right, so it says, have there been any relationship identified between molecular mechanisms of circadian rhythm such as clock and BMAL and A beta or tau? So I was involved in a study led by Eric Music and Jerry Kress. We published in animals where we found that if you delete BMAL from an amyloid model, that does increase amyloid pathology, though the data would suggest that it may be because of BMAL's disruption of the sleep-wake cycle more so than the circadian disruption, because when you only if you disrupt BMAL only in the suprachiasmatic nucleus, but not the rest of the cells in the brain, you still get that effect. But that is something that Eric's lab is actively pursuing more now.
I'm going to say, skip my question, go right to Vikram's, which is a really interesting question, David.
Okay. So Vikram, hi Vikram, said, would you postulate that each individual has a defined amount of wake time in a lifetime before developing AD? That is an interesting question. So one of the ways I've been thinking about this, that each person's brain probably has an optimal amount of sleep that it wants to undergo. And that probably is regulated by many genetic and other factors. And I wonder whether if you get away from what that optimum is for that person, whether that might lead to what you're asking about, Vikram, as opposed, in other words, it's not the absolute amount across everybody, but it's more the absolute amount for that person's brain that's probably what's at stake. But that's something that we'd like to somehow figure out a way to test. Let's see. There's a question from Syed, who says, it lends credence to Tononi's view of proposed slow wave sleep, which is synaptic downregulation rather than strengthening, isn't it? Yeah, actually, I've quoted Tononi's papers extensively in our work because I think his series on synaptic sleep homeostasis may fit very well into some of the findings that we have for these proteins that we're studying in relation to disease.
And let's see, you have one from Dr. Kaplish. It is clear the external stimulus to cause fragmentation leads to accumulation. Is there any literature on internal stimuli that lead to sleep disruption, such as breathing issues or periodic leg movements or hypoxemia?
Yeah, there are some groups that we haven't, well, there's only one paper that we've done to address this by Yoel Ju. There's other groups that have tried to address it as well. So In one human study, what Yoel did was she studied humans who have different levels of sleep apnea or not. And she tried to understand whether, at least for the A beta peptide, if you measured it, whether that was influenced by the degree of sleep apnea. And then if you treated the sleep apnea, did that improve, at least in this case, the levels of the A beta peptide? And there was basically a weird effect where, at least in CSF, we hypothesized that if you have more sleep apnea, you're obviously sleeping less, you have disrupted sleep, that A beta peptide would be higher. But in fact, we found it was lower. But it turns out that when we measured a whole host of other proteins in CSF in those who had sleep apnea, that all the proteins that we measured in CSF were lower. And we wondered, if we postulated that the reason for that might be that you are disrupting, when you have this altered breathing, it alters pressure in the brain. You have a lot of valsalvaing going on when you're having multiple sleep apneas every hour, and that might prevent the flow from brain down to the lumbar space. So that's something we need to further pursue by studying this phenomenon more dynamically, not at just one time point with a single spinal tap. Let's see. I think you want to ask some of these, or let's see. Gmo asks, since we all have different phase. Yes, to Vikram's question, since we all have different phase angles of circadian entrainment. That's an interesting comment. And Jack Parent said, if people looked at secretion of tau A beta during up and down states of neurons. It's a really good question. The problem is it's hard to do that because of the technique of measuring the A beta peptide. You can't, the most rapidly you can measure it is a technique developed by John Cerrito, where he puts a small probe in the brain that's coated with an antibody so that you can electrochemically detect it. But even that, you can only detect it about every minute or two. It's not rapid enough to really measure it with these rapid changes in activity that we'd like to be able to do.
All right, I think we should take time for one more question, maybe Dr. Feldman's, and call it a day.
Yeah, so Eva asked a good question about the Lancet Commission just published 12 reversible factors for AD, but sleep is not one of the factors. Seems like an obvious one to include. Do you know why it was not part of the recommendations? No, I don't know why it's not, but I mean, a lot of people are certainly that are just advocating for better potential lifestyle changes that could influence dementia and Alzheimer's disease. That is definitely one many people are now putting in there, but I'm not sure why they didn't do that. It seems to me something that there's certainly enough circumstantial evidence to mention it as, and for all the other reasons that you want to get good sleep as well.
All right, so I think we're going to give you a virtual round of applause. Stellar, very stimulating talk. It's going to give us a lot to think about. Thank you so much, and congratulations on being this year's recipient of the Michael S. Aldrich Commemorative Lectureship Award.
Well, thanks so much.